Hello again. Now, the importance of this slide is the previous experiment shows that for bacteria, the genetic material is DNA. But what about viruses? So Hershey and Chase, they did experiments based on viruses. What's important in this, in order for you to understand this experiment, you need to understand a few things. You need to understand how a bacteriophage is a virus that attacks bacteria. How does it actually infect bacteria? How does it attack bacteria? So what it does is that the virus will land on the bacteria, and whatever it will insert is genetic material, and then this genetic material will then be used by the bacteria. The bacteria is tricked into using this genetic material from the virus to make more viruses, and after a while it takes control of the whole of the bacterial replication process, makes more viruses, and then it will the bacteria will die. So this is what they do. So the key point here is the fact that they want to know what part of the virus is inserted into the cells, right? So that because whatever is inserted must be the genetic material. So they devised this experiment, right? Now the other technology that allowed this experiment to be successfully giving you the expected result is that there's the fact that they can label differently the DNA. So a virus is a simple molecule. It has a coat protein, and inside is the nucleic acid. Now they were able to create different strains that carries different radioisotopes. These viruses were grown in a media that contains radioactive phosphorus, and what you will get is viruses in which the DNA is actually labeled, right? Is actually labeled with radioactive. Or if they grow if they allow another strain of bacteria, another strain of virus to infect a bacteria, and then they grow this bacteria in the presence of radioactive sulfur 35S, and what you get is actually a protein that is a virus that has a protein that has been differentially coated with radioactive. So you have two types of viruses. Here it's shown here is that this particular virus has got its DNA labeled with 32P phosphorus, radioactive isotope, and then you have this virus. Why is labeled red? Because the coat is actually labeled with radioactive isotope. So what happens is that they allow this to infect the bacteria. Now, why do they need a blender? Is because they want to interrupt the infection process. What they want to do is they want to just see what gets inserted into the bacteria, and then they want to stop it. So that's why they put it in a warring blender. So that happens, I think, after about 10 seconds of allowing the virus to attack. So once they have that, so they put it in a warring blender, and then they isolate it out. Now, so what happens then is that they do centrifugation. What is centrifugation? For those of you who are not familiar with centrifugation, it's a mechanism where you actually spin the tube that contains the bacteria and the viruses, and then so what happens is that because of the centrifugal force, the heavy stuff will be embedded towards the end of the tube, and then lighter stuff will be at the top. All right. So basically, because bacterial cells are much larger, so all will be sedimented at the bottom of the tube, and then now you have two experiments here. You will find that for this particular experiment, a lot of the radioactivity is at the bottom. Right, a lot of the activity is at the bottom of the tube where the bacterial cells lie. When compared to the other set of experiment, they found that for this experiment, the radioactivity actually remains in the supernatant. All right. Now, what happens here is that in this experiment, the DNA has actually been inserted into the bacteria. Remember, the DNA is radioactive in this experimental set, so that's why the bacterial layer is radioactive. Whereas for this part, also the DNA is also inserted inside the cells, but the radioactive part is actually in the virus's coat protein. Coat protein do not enter the bacteria, so because it's very light, it remains in the supernatant. It doesn't get sedimented to the bottom. So that's why for this experiment, the radioactivity remains in the supernatant. So you know, this again is a brilliant experiment to show that again, DNA material the genetic material needs to be inserted into the bacteria for it to function. And in this, they were able to show that only DNA that is inserted and the protein coat does not get inserted. For that, they show evidence that for viruses, the DNA material is the genetic material, and similar case with bacteria. This experiment, on the other hand, talks about viruses that do not have DNA. So there are various types of viruses. The one that Hershey and Chase did was a virus that uses DNA. Then the question comes that, you know, what about viruses that has RNA as a genetic material? So I'll explain this in the next video. Bye-bye.